Hello, everyone. I am Carrie Blood, and I am the Community Engagement Director with Kestrel Land Trust. And I want to welcome you all to this presentation today. As we all know, our rapidly warming planet is the biggest challenge humanity has ever faced, and we need to make use of all the solutions and tools we can to address it. And protecting forests and other undeveloped lands is one of those solutions, which is why Kestrel's mission to conserve and care for these lands is really more important than ever. And it's why we are hosting this speaker series. And I think we're ready to introduce our speaker, Dr. Peter Curtis. So Peter lives in Northampton, and he is Professor Emeritus of Plant Ecology at Ohio State University, where he taught courses in botany, ecology, ethnobotany, and climate change biology. He has published widely in the area of plant and ecosystem responses to climate change, and we are so pleased to have him with us tonight to share his knowledge. So welcome, Peter, and I'd like to turn it over to you now.
Thank you, Carrie. Can you hear me all right?
Yes, we can.
Okay, well, thanks everybody for coming today to hear this presentation. Let me, I'm going to share my screen and we can get into this. You may see everybody. That look okay to everybody? Look good to you, Carrie? Give me thumbs up. So yes, well, welcome. You know, it's a warm day in April, and I was reflecting on this today as we think about the topic of today's talk on climate change and the pace of seasons in the Pioneer Valley. You know, is it okay to enjoy this kind of warm weather? And I am always feel a little guilty. Is this a sign, sort of an ominous sign of things to come? But we do anyway. Things are popping out. The flowers are coming on strong, and this is all very sort of sets up this presentation really nicely. So I'm going to get started. I want to just spend a minute or two just giving folks a bit of idea about my background. So as Carrie mentioned, I'm a Emeritus Professor of Plant Ecology at Ohio State. I'm a botanist by training. I've most of my research for the last couple of decades is focused on forest ecology and how carbon is taken up by forests and stored, and particularly how that might be affected as forests age. And so this is an interesting topic in forestry and in ecology. Much of the work that I've done has been centered in the northern Great Lakes area, particularly at a biological station here at the tip of the mid and northern lower Michigan, where we put up equipment like this tower so that we can measure CO2 as it comes in and out of the canopy, allowing us to create diagrams like the one here on the right, quantifying how much carbon is taken up in photosynthesis, how much is lost in respiration, and the carbon that's stored, where does it go in leaves and the stem and roots and soil and so forth. So that's been the bulk of my research, but as sort of part of that, we've also been very interested in how climate change and climate in general affects the growing season length. And this is part of phenology. This is something I'm going to be talking about. So that's been one area of research of ours. And I'll mention that towards the end, but also one of my students in particular, Dr. Kellen Callinger, was interested in how climate affects different spring wildflowers in terms of their time and flowering, and how much variability there is in that response. And so that's something that we're going to be spending a bit of time on today as well. So that's a little bit of my background. Currently a resident in Northampton, having left Ohio a couple of years ago. So where's this presentation going? What am I going to be covering? What will we be considering? Well, first I want to talk about what is phenology, which is the science of appearances, the same root term as phenomenon or phenotype. I want to go then into the discussion about how climate change relates to phenology, how those two things interact, for example. I want to talk about the ecological consequences of changes in phenology, particularly with plants, but we're going to talk a little bit about animals as well. And I want to conclude with a sort of presentation about how any of us can be involved and get involved participate in monitoring phenology around where we live, in this case, in the Pioneer Valley. So there's lots of opportunities for something we refer to as citizen science in monitoring and thinking about and tracking phenology. So that's pretty much where we're going to be going. Oops, let's do this. So phenology, and I always have to say this, not to be confused with phrenology, which of course is the 19th century study of bumps on people's heads. This is phenology, and it's fundamentally or broadly the study of the timing of key life events. It's biological processes. So it might be the time of flowering, in this case, the Dutchman's breeches there in the photograph, when animals may emerge from hibernation or go into reproductive status, nice marmot photograph. The timing of leaf drop in the fall is another phenological event. Bird migrations are a classic, and when they might arrive on their breeding grounds is a phenological event. So the timing of these life events is all swept up in the science of phenology. We know now that because, and I'll talk about this in a second, because phenology is so intimately tied to biological indicators in the environment or in weather in many cases, not always, but this is a sensitive biological indicator of climate change. Unlike weather patterns, for example, this is something in biology that we know is sensitive to the ongoing process of climate change. And moreover, this is something that we're seeing now around the world. So globally, worldwide, it's becoming increasingly well documented that we're seeing shifts in the timing of key life events and phenology in both animals as well as plants. So what are some phenological events? What are some examples of this? And I'm going to start with plants. And this diagram, I enjoy this diagram because it sort of shows how plant phenology is connected to everything else. And this is sort of the classic ecology diagram where something is in the middle, in this case, phenology. And so some of the classic plant responses that we think about are bud burst or leaf emergence in the spring. This would be in the temperate zone. Flowering, which can, of course, happen any time of the year, spring, summer, fall, and again in the temperate zone among plants, or leaf drop, typically occurring in the fall. So these are all phenological events for plants. And as you can see in this diagram, these are linked with other kinds of processes. Pollination, for example, something we're going to talk about with flowering. So then the pollinators that might be responsible for that. When leaves drop, the canopy changes color and it has different radiative properties. And so that's one of the follow-on events. And phenology is affected by different things as well. Weather is something we're going to be talking about, but other things as well as evolutionary processes, the structure of the community that plants and animals find themselves in. So highly complicated in that sense. Some examples of bird and amphibian responses, things that are going on right now. So we're sort of in the thick of it. When migratory birds arrive, wherever we see them as part of their migration, or maybe where they're going to be breeding, when they begin nesting behavior or egg laying is a phenological event. Frog vocalizations, I think, is just tailing off now. Of course, the spring peepers, the tree frogs have been out there busy vocalizing. The timing of that is a phenological event for an animal. Insect responses, very important, and for many of the things that we're already talked about here and the plants and animals. But when insects emerge, when they may arrive if they're migratory, monarch butterflies, example. Very important for forest health is the number of life cycles per year that insects can achieve. In many cases now, where because of the onset of winter or the late springs, the insects might have only had one life cycle in a year. Now they're able to do two with warming temperatures. So these are all examples of phenological events, from plants to birds, amphibians, and insects. So what are the factors out there in the environment that influence these? How do we understand these in terms of their responses to the environment? Well, these are sort of the big three that I have illustrated here right now. One that is an important driver for certainly plants and animals is the length of the day or the night, and something in biology we refer to as the photoperiod. So you can imagine this is a pretty good indicator of the time of year in the temperate zone with the photoperiod or day length lengthening in the spring and then shortening in the fall. And so both plants and animals use that photoperiod in their timing, in their determining their phenological behavior. But as part of something that is a strong modifier of that is temperature. So you can imagine that even though it may feel like mid-April, if it's a cold spring, that might allow an animal or a plant to delay its flowering or its emergence. And likewise, if it's a warm spring, it might accelerate that. So temperature is an important modifier for many species, many organisms, in terms of their photoperiod response. Precipitation, again, is something that's very important. And this is particularly true out west or in arid environments. And I think some of you are certainly familiar with the desert bloom, which is so closely tied to how much rain fell in previous months, if you're out in the desert, for example. So photoperiod, temperature, precipitation, very important environmental drivers, environmental influences on phenological processes. But there are others as well, and they're less well studied in many cases, but I just want to mention them because we need to understand those as we think about the variability in responses that we see out there in nature. So for example, the timing of snowmelt is very important for many early flowering species. And this is particularly true in alpine areas, or in a similar vein, ice out, if for aquatic organisms in lakes, for example, or for that matter, in oceans as well. So those would be important timers or influences on phenology. Disturbance events, fire is an important one, again, and often in more arid environments, drier environments. But there may be other things as well, ice storms and that sort of thing that may open up, may change the local environment. That can influence phenological processes. Genetics plays an important role. It's something we're increasingly appreciating. That can be both between species, and something we're going to see shortly is that there's quite a bit of variability among different species and how they respond to the same environmental cues, but there also may be variability within species. So some individuals might respond more strongly to, say, temperature than other individuals. And finally, I do want to mention the community. So this might be the plant or animal community, the symbionts that an organism might be interacting with, pollinators and their hosts, competitors in the environment. The makeup of the local community, plant and animal community, may also influence the timing of these events. So we have to keep all of these in mind, but the one that we're going to focus on, partly because it's the best studied, particularly as it relates to climate change, is temperature, and particularly the temperature in the winter and the spring as it influences spring phenology in both plants and animals. So that's that, but keeping all of these in mind as we go forward. So how much has temperature changed in New England? And I'm sure I don't have to convince anybody that we're in the middle of rapid climate change. But one thing that not everybody appreciates, that in New England is changing more rapidly than virtually any place else in the US outside of Alaska. And so this is a map I taken from the EPA website, quite recent information looking at the rate of temperature change between 1901 and 2020. And the degree of redness is an indicator of how much temperature is rising. And in some cases, there's a little bit of cooling down here in the south. But as you can see, New England is the deepest red. And this is probably due to a number of things. And I'm not going to get into this in any detail, but changes in the ocean circulation, in particularly in the Western Atlantic, is caught in the warming there of those ocean waters is being reflected in warming in the land surface as well. So we're really in the thick of it here in New England, very dramatic and rising temperature, more so than we've seen just about any place else. So that's sobering. Well, how much has temperature actually changed? And we'll zoom in here a little bit on Massachusetts. And there's a number of ways that we can look at historic temperature, but we're fortunate in the United States and also in New England that we've been making instrument measurements of temperature for quite a long time. And there's a series of what we now refer to as the historic climate stations or historic climate network throughout the US. And this illustration here is from a recent paper, just showing you these little pinpoints of where these historic climate stations are located. So you can use those climate records with appropriate adjustments for changes in landscape, for example, to analyze how much temperature has actually changed. And so what we find in Massachusetts, and that's what this figure here is, and the orange line is the average temperature, and the blue line is our minimum temperatures, and the gray line are maximum temperatures through the years starting in the 1900s through to the 2010s. And the result here, the conclusion is that temperatures increased about 2 degrees centigrade, 3.6 degrees Fahrenheit since 1900. So full 2 degrees centigrade increase over that time period. The minimum temperatures have increased even more, and that's actually an important story here, is that winter temperatures seem to be responding more to climate change than are, say, summer temperatures or fall temperatures. So minimum temperatures have increased almost 2.5 degrees. So clearly temperature has increased 2 degrees since 1900. We'll come back to that in a bit as well, with winter temperatures increasing even more. Well, what has this meant for spring, the pace of life in the spring in Pioneer Valley? Has this century of warming affected spring wildflowers in Massachusetts? Well, we're fortunate in this state to have a great record that stems back to the work of Henry David Thoreau, a celebrated, of course, author and naturalist. And in Concord, when he was there, he was an assiduous observer of nature, as I think where most of us are familiar, keeping notes of his forays into the woods. And in what is, I understand to be notoriously hard to read handwriting, but it's been deciphered. And he recorded first flowering dates for over 500 plant species blooming in Concord when he was there between 1852 and 1858. So we have this very detailed record, 500 plant species, first flowering dates in the 1850s. Well, then he stops. But fortunately for us, his effort was continued by a big fan of his, a fellow by the name of Alfred Hosmer, who continued these detailed records for as many plant species. It wasn't 500, but for many of the same species between about 1872 and 1902. So for 30 years, Hosmer recorded first flowering dates. So we have this data set from the 1850s and then the 1870s to 1902. And then it sort of goes dormant until a contemporary group of researchers, two people from Boston University, Richard Premack and his student Abraham Miller Rushing, who really brought this data set to light and took it upon themselves to go back to Concord and record first flowering dates for as many plant species that were also seen by Thoreau and Hosmer as they could. So this now is a famous data set, and they did a great job with this. It made a big impact. And I want to look at those data to see what they did and what we can learn from them. And so that's what we're showing here. So just I'm going to sort of step us through this diagram from their publication, one of their key publications back in 2008. So what they did is they looked at, of those 500 species, they had good data for about 43 species where Thoreau had looked at them, Hosmer, and they could find them as well. And they averaged all those 43 species. They did some statistical averaging, so coming up with an average flowering date for that year. And that's actually what's graphed here. And overall, they then said, okay, so in Thoreau's time, on average of those 6 years that he was making observations, the average flowering date was about May 14th. And we go back to Hosmer, it's a little bit earlier. And by the time Miller, Rushing, and Premack published this paper, or actually in 2005, 2006, that average spring flowering time had advanced about a week. So up to from about 14th of May to the 7th of May. So one thing we can conclude from this, or they concluded, is that indeed flowering has advanced a full week on average. Now, there's a lot of variability there. And some species that they were looking at have advanced considerably more than a week. So for example, high bush blueberry, 21 days early, 3 weeks earlier than when Thoreau was making his observations. Yellow wood sorrel, a month earlier. And now, not all of that is directly attributable to temperature change, but there's a lot of variability. But they also flag what they called indicator species that seem to be particularly sensitive to temperature. That seemed to explain a lot of their earlier flowering behavior. And these are two of the species that they identified. Blue flag iris, very closely coupled with temperature, about 4 days in advance, earlier per degree centigrade. So maybe 8 days over those two degrees that we've warmed. Canada Mayflower, another species that shows a strong temperature response. But just again, to keep in mind, other things are changing over this 150, 160 year time frame in addition to temperature. So those are really important data, and they tell us something about what's going on in Massachusetts. But one of the questions we also could pose is, can we get, these data are, there's a group at 1850s, another group around 1900, and then we have a big gap until 2000s. Is there a way that we can get a more complete record of past flowering dates? And Premack and Miller Rushing have done this, but as well as my student, Kellen Callinger. And the solution to that has been to make use of this great collection of something we refer to as herbarium specimens. Now, I'm aware of not everybody knows what a herbarium specimen is, so allow me to just briefly describe how this was done. And I got this great picture of Alice Eastwood, who is a curator of botany, remarkable botanist, remarkable scientist. At the turn of the century, about 1900, she was curator in the 1890s at the California Academy of Sciences. But this is a great illustration of how botanists worked in the field for decades, centuries really, is that we would go out in the field, not always in such a nice hat, but carrying a plant press. And as you're collecting plants, we didn't have cameras and other ways of recording images that we have now, much less collecting is done now. And those plants that you would collect would be very carefully pressed, put into your plant press between absorbent paper. You'd bring that back to the herbarium, carefully tape it to the herbarium sheet, label it, and this then goes into cabinets and becomes this record of that plant specimen. And you can see, in this case, Dutchman's breeches, whether it was flowering or not, and perhaps the degree of flowering. That's how we came upon all these plant specimens, these herbarium specimens. And there are thousands of them, actually millions of specimens in various herbaria that have been collected over decades, in some cases centuries, depending on where you're going. They contain a lot of information, of course, the plant itself, what it looks like, where and when it was collected. And we can infer from that, not so much the first flowering date, but peak flowering. So that can give us this similar sort of information. Now, I note down here, I'm going to be talking about information that was collected from the Ohio State University Herbarium, which we thought was pretty big, 500,000 specimens, that's a lot, focusing really on Ohio. I have to mention, though, the Harvard Herbarium has 10 times as many, 5 million specimens. New York Botanical Garden Herbarium, I think, is about the biggest in the US at 7 million specimens and growing. But in this case, my student, Kellen Callinger-Yoke, took this task on as part of her dissertation, one of the first, and she was following in the footsteps of a few others. But I want to show you some of that information, because it's so important as we sort of think about how different species are reacting to climate change. So we're going to see a little bit more data here, but bear with me. I think it's interesting. So this is how we calculate something that we refer to as phenological sensitivity. So how sensitive is a plant in terms of its flowering to temperature? And we can go back and look at those herbarium records and pull out specimens which are in their peak flowering time, and we note the year. And if we have a historic temperature record, we can pair that specimen with a temperature reading. So in the case of pink lady slipper orchid from Kellen's data set, she found 29 specimens in peak flowering that had been collected over about 90 years, from 1898 to 1988. And she just arrayed them according to the day of year that they were flowering. So they varied from early April or mid-April to late June. And this is across Ohio, certain amount of variability. And I just want to note just some of these data points when they were collected, just so that you have an idea of how this is done. These aren't arranged chronologically, they're arranged according to the temperature, the spring temperature preceding when they flowered. It's important to note that. So on the x-axis here is the spring temperature, which typically was the average temperature of February, March, April, and May. The month of flowering, the 3 months prior to that. So plants sort of sensing that environment. So for example, in 1907, a very cool spring, fairly late flowering. 1988 over here, quite a warm spring, a much earlier flowering. So we have a negative slope to this line. That's our phenological sensitivity. Now, something funny is going on in 1901. We don't know what it was. So it was sort of an average spring temperature, but for some reason, the pink lady slippers in Ohio weren't flowering until late in June, very late, even though it wasn't even all that cold. So we don't know. That's part of the variability. Maybe something else was going on. So that would be the sensitivity of pink lady slipper orchid to temperature, about 3.5 days per degree centigrade, really just the slope of that line. Everybody with me? If you're completely confused, send a chat message and Carrie will flag me. We'll go back to this, but that's just one example. Here's another example, same analysis, but in this case was flowering dogwood, more specimens collected. It's all plotted down here over a slightly longer time period, well over 100 years, 111 years. And in this case, you can see the slope is not quite as steep. So somewhat less sensitive to temperature, advancing its flowering only about 2.5 days, 2.3 days per degree centigrade. So this is how we go back and use these herbarium records, pair the time that we could see that they were flowering with our best measurement of temperature at that time, the closest temperature record we could find. There's invariably going to be some scatter in that. And we can essentially analyze lots of different species of how sensitive they appear to have been to temperature. So that's what we're going to see now. This is not going to show you too much more complicated data like this, but bear with me because it really shows you how much variability there is out there, and not everybody in the landscape is going to be responding the same way. So Kellen was able to analyze 140 responses for 140 species. You just line them up here on this, just 1 through 140. And this is their sensitivity, that flowering shift, the slope of that line, really, how steep it was. Everything to the left of the 0 shows species which advanced their flowering as it got warmer. So they flowered earlier. And there's quite a few around 0. So that would be no change. So if they flowered May 10th, and they pretty much flowered May 10th, whether it was a warm or a cool spring, for example. There are a few cases where plants actually flowered later when it was warmer. So overall, of these 140 species, about half, 66 species, significantly advanced their flowering with increased temperature. So they flowered earlier in the year. So, on average, 3.7 days per degree centigrade. About half showed no significant change in flowering. So they're pretty insensitive, lots of variability. They may be responding to other things, pollinators, disturbance, something else, precipitation perhaps. And then a few, 2, showed significantly delayed flowering with increasing temperature. These are typically plants which flower later in the spring or in fact in early summer, less sensitivity to temperature overall. So lots of variability among species, and that's an important message. Here's another, just a graphic, less data intensive way of looking at this. So we can look down here with the seasons we have an early spring, as the seasons are getting earlier here, say an earlier spring. Some species, wild indigo here, shows a very strong shift in flowering, so that'll track that increase in temperature. Mayapple, as an example, has a little bit of a shift, maybe a half a degree, a day or a half a day per degree centigrade. And then there are a whole bunch of species, shooting star as an example, which just pretty much flower the same time regardless if it's a cool or a warm spring. So considerable amount of variation. So why does any of this matter ecologically? Well, a number of reasons. There are potential risks or benefits of different phenological responses. So if you're flowering early or not all of these responses are going to be equally successful in terms of producing seeds or producing offspring if you're a bird or a mammal, for example, and you're tracking or not tracking temperature or climate change. And for that reason, because there are both risks and benefits, and there's going to be uneven benefits, or not everybody's going to be equally successful, there could be significant impacts on biodiversity. In other words, winners and losers in their response, their phenological response, and hence their reproductive output. That's what we often focus on. So what are some examples of that? So one that is discussed a lot as a possible risk is something we call a mismatch, or a trophic mismatch, or pollinator mismatches. So if you had a certain circumstance where normally in a normal year and within the normal range of temperature variation, the bees and the flowers would be emerging more or less at the same time, and you have bees visiting flowers and pollinating them, and fruit set occurring because of that. But for example, if the flower responds more strongly to temperature or climate change than does the bee, you then have this offset. So the flowers are blooming, there's no bees around, and they don't receive pollinator service, and they may not set any seed. Similarly, the bees may emerge and not find the resources that they're expecting in terms of pollen or nectar. So that's a possible mismatch. And there's a little bit of evidence for that. It's not easy to tease out from other ecological factors. But on the flip side, there may be some benefits to separating yourself from the gang. So for example, here's a normal flowering. You might see a bunch of species flowering at once. So violets and trilliums and spring beauty. And there's something we call pollinator competition. There may not be that many bees out. And so you're competing with your neighbors for access to those pollinators. But imagine the situation where trillium is responding more strongly than spring beauty or violets. And it now enjoys, assuming that there are still pollinators around, enjoys less competition, maybe greater access to pollinators. So that's one possible benefit. Another possible risk, frost damage. This is a case where there may be a warm spring. Some species really react strongly to that, but it was sort of a false alarm. And in this case, these are just pictures that Kellen took from 2007, where, oops, go back. They're April 3rd, maples are flowering or fruiting. There was a hard frost on April 8th, I think. And the consequence of that was the flowers died, and the flowers and the fruits of these silver maple and Norway maple were killed. So a species which didn't respond so strongly to that early, that spring warming would not have suffered that consequence, this frost damage. So increased likelihood of frost damage for early flowering species, or ones that track temperature more precisely. A benefit, maybe a longer growing season for lake robin, our trillium or red trillium. So moderate temperatures, spring occurs over a certain period. Maybe with warming spring, you have a longer period where trillium or other species may be able to enjoy photosynthesis or access to pollinators. So possible benefits as well. So just to emphasize, there's a lot of variation, and we're thinking about possible risks and benefits to these different responses to climate change. We know there's a lot of variation. We're still trying to uncover who may benefit and who may not. Let's shift gears here a little bit, see where we are. Birds. So it's complicated, but I do want to spend a little bit of time talking about birds because it's been a source of inspiration for people for a long time. We all look forward to certain species arriving in the spring. My grandmother made notes on her bird book, which we found in her library, when she would observe the first robin coming. This was up in Maine, for example. And for this record, a great record, we go back again to Thoreau, who was in addition to a good botanist, he was a good field ornithologist and made detailed records of when birds arrived in Concord, Mass. His observations were followed by a renowned ornithologist at Harvard, William Brewster, who made observations 1900 and 1919. And then we're really fortunate, and this is one of these examples of citizen science where no one was really expecting this to be relevant, but a schools teacher, Rosita Corey from Concord, very skilled amateur ornithologist, made observations from 1956 to 73, took a break, came back in 1988, and then until shortly before her death around in 2007. And another student from Boston University, Elizabeth Elwood, went back to those records to try to do the same thing that they did with the plants. If we looked at how many species they had in common with these different observations, and Elwood was able to look at 22 bird species when they arrived in Concord, Mass. Between 1851 and 2007. And the bottom line is complicated, and then there were no overall clear trends. But I really want to highlight a couple of birds over this 157 years. But there's a number of reasons why birds are complicated in this regard. A big one, and this is a worry for a number of reasons, is that the timing of arrival and how we observe it may be in part determined by whether these bird populations are increasing or declining. And many of our migratory bird species are declining in abundance. So if there are fewer birds coming, there's greater likelihood that you'll miss them. So it's very possible that a late arrival is more of an indication of declining population trends than anything else. So that's a worry. Where birds are overwintering, if they're migrating from Costa Rica versus from Louisiana, that makes a difference on how closely we might expect their behavior to be influenced by something like temperature, as opposed to big weather patterns. For example, if they have to cross the Gulf of Mexico. So there's a variety of factors that make this more complicated. But nonetheless, there are some species which are responding to temperature. I'm just going to highlight some of those. And I don't know how well you can read this table, but look at warbling vireo, cute little vireo. It normally comes around May 10th in Concord, can arrive as late as June 10th, a whole month later, or quite a bit, a couple of weeks earlier into third week of April. So quite a bit of variability. But when Elwood plotted all those numbers, it became clear that this bird was responding positively to temperature, about 2 days per degree centigrade. So appears to be coming earlier. On the other hand, here's the wood thrush, one of my favorite birds in the spring. Usually comes around the same time, 8th of May. It can be as late as the 19th of May over this time period, or 26th of April on the early side. It seems to be responding in the opposite fashion to temperature. In other words, coming later in warmer years. We don't really know why. And lastly, my last example here is the Baltimore oriole, another flashy bird that we'll see in the spring. And this is usually comes around the same time, 7th of May, can't wait. Could be later, could be earlier, considerably earlier, but doesn't seem to be strongly connected to temperature, maybe a half a day per degree centigrade. So what's behind all this variation? Ornithologists, I hope, are working on, but at least for this data set, no clear trend, this particular data set. So those are some of the bird data. I want to then now, we've been thinking about sort of small scale individual species, but I'm an ecosystem ecologist and have spent a lot of time thinking about forests and their canopies and how they're put together. And an exciting development has been a proliferation of something we call the phenocam, or phenological camera systems. Of course, there's cameras. Everybody seems to have a wildlife camera out there now, and we can track things very carefully. Same thing is true with phenologists. And there's a group that was now out of Northern Arizona University who's coordinated all of this. But for many of us who have built canopy towers, we can now put a camera at the top of them and make a continuous recording, just looking down at the canopy. And you can take those images and look at greenness and redness and really figure out pretty carefully, pretty precisely when the green up occurred and when leaf coloration and leaf fall happened. I'm going to show you some of that information in a second. But so that's from these towers, and we had one on the tower that I developed in Michigan, and there's some nearby here too as well. I'll show you those in a second. But we can also see this stuff from space now. Of course, with satellites, almost anything is possible. And I just have to show you this little GIF. Because there's a number of satellites up there that can detect vegetation changes from space. And it's really becomes quite powerful when we combine these phenocams with satellites. But this literally is the breathing of the Earth. So the green up in the spring, leaf fall and ice coverage in the winter. This is very closely coupled to CO2 oscillations, oxygen oscillations in the atmosphere. So these have big effects. These phenological processes have big effects on climate and literally can be seen from space. So that's pretty exciting for us who study larger scale processes. But I want to just drill down a little bit into those data in the phenocam network. So there's quite a few of them. These are the ones that are scattered around the US. The green ones are all active. This one right here in northern lower Michigan is the one on my tower. You can see we have a bunch in Massachusetts. In fact, this is the group. There's about a dozen in Massachusetts of these cameras, these phenocams, sitting on towers, beaming their data to Northern Arizona University. And one of those is at the Smith College McLeish Field Station. And I just did a screenshot of the images coming from the McLeish phenocam, looking right down at basically at the canopy. So this image was grabbed on February 21st. You see the snow on the ground. That's pretty typical February. Here's one for March 26th. And there's no snow on the ground, but there's no leaves on the trees either. And I grabbed one more image just today. And if you squint, if you're looking at this carefully, you can see some red in there. Those must be red maples or maybe in that canopy. I haven't been there. So spring is on the way. And those data are, again, being assimilated along with data from all these cameras mounted on these canopy towers. So what are they telling us? What do we know about the growing season length? Well, it's getting longer, perhaps not surprising with the information we've just given you, but it actually works at both ends. So globally, so there have been people, cameras are up in lots of different places in the temperate zone. Growing season is lengthening about 3 to 4 days per decade. So even since the 2000s, our growing season in temperate forests has increased overall on average, maybe a week or so. This is due both to earlier leaf out, but also later leaf fall as well. So it's working both ends of that. So the green canopy is out there for a little bit longer. So one of the important questions for us, ecosystem people, is, does this allow more carbon uptake and perhaps carbon storage? Is this a potential break on some aspects of climate change, particularly CO2 in the environment? And indeed, there's pretty good evidence at this point that there is more photosynthesis happening globally, in part due to this lengthening of the growing season. There are other factors as well, particularly rising CO2. Let's see, but it's, as with most things, it's complicated. Different areas show different sensitivities for these phenological events to temperature when we're looking at it at the canopy. This is a very recent paper looking at the sensitivity of the green up stage. So this over here across the US from phenocam data. And what these authors found was that in the heat map here, the degree of red shows the greater sensitivity. So from 3 to 4.2 different shades of red. And what they found was that the southeastern US is at present more sensitive in terms of the green up than is New England, which is still advancing, but not as much as the Southeast. So we see a sort of dynamic response. And that may be because spring is typically somewhat longer in the Southeast. And these authors also project that in the future, this color map will change, and that as the climate warms, that New England will sort of essentially be a similar shade of red, show more sensitivity to temperature increases. So yes, indeed, the seasons are getting longer. Green up is happening earlier, leaf fall, and oftentimes later. Okay, to wrap up, getting involved in doing this sort of thing, getting involved with monitoring phenology, loads of fun. And there's great opportunities. And I want to give a pitch now for getting involved with this. There are both national and local organizations for citizen science contributions to our understanding of phenology. There's a National Phenology Network, which I'm going to have more to say about, the NPN. There's more local efforts as well. This is one, Project Budburst from the Chicago Botanic Gardens, and I'm sure there are others as well. But the NPN, the National Phenology Network, has a great platform for citizen science. It's called Nature's Notebook. It's got a bunch of people working on this. This is just, again, a shot from their webpage. Nature's Notebook here. They do forecasts and other sorts of things. But it's a great platform for volunteer scientists, citizen scientists, for educators, land managers, reporters, researchers, and others as well. So that's a really interesting and really beneficial platform. And it's the one that I use for recording, going out and recording phenological observations. Many tools that different individuals or groups, educators can use. So again, just a screenshot here. It's not just for plants. It's a platform for recording animal observations as well, insects, birds, amphibians. And as with everything these days, there's an app. They've put quite a bit of development into this app. So when I started this, I would write everything down, and I'm out in the field and transfer that to the online, my computer platform. You can do all that from your smartphone now. And so that's an opportunity. And one of the things, well, to back up, one of the things for me that's so enticing about this is it gets me to slow down when I'm in the field and really pay attention to what's going on, sort of at a granular level. It's like slowing down and smelling the roses. This is more like slowing down and looking at the bloodroot a little more carefully, or looking at the trout lily, what's coming up when, and looking at that year after year. And that can be really a lot of fun. A whole bunch of researchers, people in the field have put together coordinated campaigns, for example, that you can participate in. One's called Nectar Connectors. It focuses on the health of monarch butterflies and recording the flowering of the plants which monarchs use to feed on, for example, before they lay on milkweeds. So there's Nectar Connectors for monarch conservation. Green Wave, sort of what we've already been talking about. Maple, oaks, and poplars. Fall color, helping us better understand what's going on with that whole canopy, that whole growing season. And one that I'm really excited about and I'm hoping to participate in is called Quercus Quest. Quercus is the genus of oaks. This is being spearheaded by some really great ecologists at the University of Minnesota, but they have a hypothesis about hybridization in oaks. And as some of you may be aware, the red oaks can hybridize with each other. In some cases, the white oak groups can hybridize, but they have to be flowering at the same time. And that's influenced by their phenological response. So these researchers have a hypothesis about how climate change might influence the likelihood of hybridization in these oaks. And they need people on the ground in different places to record flowering in these oaks. So that's kind of exciting. So if any of this piques your interest, I'm going to give one last final pitch. Join us on 3 weeks from today on May 7th for more on wildflower phenology. I'll be leading a field trip at the Dyer Conservation Area in Hadley. And we will, it'll be an opportunity to both take a wildflower walk. I mean, it's a beautiful time of year, May 7th. There'll be lots of things up. You can brush up on your wildflower ID. Plus, I'll be giving a tutorial using the Nature's Notebook app. And afterwards, we can gather at the Porter Phelps Huntington Museum to brainstorm about how we might continue this, particularly bringing in school groups or other interested folks for more sort of focused trails where we can monitor the same thing year after year. So that'll be a lot of fun, and I hope to see everybody there on May 7th. And hopefully the weather was nice. So with that, I think I'm just about out of time. And thank you for your attention, and we'll be happy to answer questions. And I also want to give a shout out to those wonderful photographers who put some of their images on the Creative Commons and let folks like me make a more colorful presentation. So with that, I will stop sharing and turn it over if there's any chats or if there's any, people can raise their hand or
So yeah, thank you so much, Peter. That was wonderful. We don't have any questions in the chat yet, but please feel free, anyone, if you would like to put a question out. We have a relatively modest sized group, so if anyone actually feels like raising their hand and putting their video on to ask a question, you can do that as well.
We can do that as well.
So yeah, please feel free.
You know, it's things are just starting to happen so quickly now out there. You know, it's one of these things where you can go out and see something new. And I have found that I've been following a skunk cabbage, you know, just down the hill from our house. And for me, I always have to go out and find the first skunk cabbage because that's the first sign of spring. But now everything's sort of piling on. The trout lily are coming up and so forth. So it's a great time of year. A little bit worrisome that it was 70 degrees the other day, but or 75, I think.
Well, at least it waited till mid-April for that to happen. Sometimes it happens a lot sooner than that.
Yeah, it does. It does.
One note about the wildflower walk at Dyer, which you mentioned, was actually only a few spots left already. So we haven't even officially announced it yet, and it's already close to full. So just a couple spots left. People are excited about that one.
Yes, so that'll be a beautiful day, I'm sure.
Yeah, definitely. So anybody? Oh, okay. Let's see.
Oh, I have a hand up from Fran. Fran, if you unmute yourself, you can just.
Hi, Peter. I'm sorry, my head is gone. Thank you so much. This was so interesting. I really appreciate it. I had a question about the fact that climate change seems to be increasing more and more rapidly. And on most of the data that we have, the time is linear. And I'm wondering if there's anything out there that gives us any indication of the fact that the time that global warming is not linear.
Right. No, right. That's a really good question. You know, I mean, you're absolutely right. It's discouraging, to say the least, that, you know, the rate of increase is increasing.
Yeah.
It's not staying the same. It's not a linear function. It's approaching, you know, more exponential than linear in many cases. Are we seeing anything any of that in these biological indicators? And that's really, I got to think about that. I mean, part of it's going to be separating signal and noise, and that's always a problem in ecology. So there's all those different things which are impacting the timing of events or an organism's behavior, whether it's a plant or an animal. So in many cases, the linear response is just the one that is sort of the best statistical fit, but it's all, and you have all these interactions, and you go back to that figure where everything's connected to everything else. And the thing that people are really concerned about now, in many cases, is not, you know, there's the individual response, but nothing exists in isolation in nature, of course. And so these pollinators, or it gets even worse because we know so much less about it, are some of the things which are going on below ground, for example, with fungi and other microbes. And so those have their own response to all of these things. And so there's that. Of course, then we have a much better idea of the way plants communicate with each other now. And so how is that being impacted? So all of that it's a great, you know, these are, in academia, we say that would make a great dissertation project, although, you know, the messier they are, the more frustrating it can be. But you make a really good point, and indeed, you know, certain things that it's more the you know, physical parameters, things like ice out or methane emissions, these things which are really troublesome, which are also, we're seeing are clearly not linear. They're you know, again, non-linear, up faster. So a very good question, though. I'm sorry I couldn't give you an example right off.
Thank you very much. That was helpful.
Anyone else have any questions? That's a great question.
Well, we can all get on with our evenings, and Thank you for your attention.